I would now like to talk about the embryology of early development in vertebrates. Looking at the adult structures of the various vertebrate groups, whether it's fish, amphibians, reptiles, birds, or mammals, you obviously see significant differences in the adult forms. However, looking at the embryology, there are significant similarities in all of these vertebrate species. The fish and the amphibians are called anamniotes, and they're called anamniotes because they must have part of their existence in water. They must reproduce in water and go back to the water because they do not have eggs which carry water in them. If you look at the reptiles, birds, and mammals, however, they can lay their eggs on land because they have their own source of water within the eggs, and they are called amniotes. Nevertheless, in spite of these differences, there are some very significant similarities, and we'll look at these similarities as we go through the vertebrate species. I would like to start with the amphibians because the amphibians have really had the most work done on them, and all of the classical early work was really done on amphibians. More recently, fish, particularly the zebrafish, have become very popular, and many of the things that were found in the amphibians have now been confirmed with zebrafish. And it's very reassuring to realize that the early work done on the amphibians has now been reinforced and confirmed in fish species as well as in birds, mammals, and some of the reptile studies. We will start with a discussion of amphibians, and in fact this lecture will be primarily about amphibians because amphibians really were the first vertebrates to serve as models of development. As we said in recent years, zebrafish have also been used extensively as amniotic vertebrates and have really confirmed many of the earlier findings for the various amphibian groups that have been looked at, primarily nectarous groups, in other words, the salamanders, as well as the ranipipians, as well as the African frog, Xenopus. As we look at early amphibian development, the field of experimental embryology was at one time dominated by these species. With their large cells and rapid development, salamander and frog embryos were wonderfully suited for transplantation experiments, for example. However, this domination ended during the early days of developmental genetics, in part because these animals undergo a long period of growth before they become fertile, and because their chromosomes are often found in several copies, precluding studies on mutagenesis. With the advent of molecular techniques, however, such as in situ hybridization, antisense oligonucleotide studies, chromatin immunological and immunoprecipitation studies, as well as other studies on proteins, researchers have returned to the study of amphibian embryos and have been able to integrate their molecular analyses with the earlier experimental findings that were published a decade or maybe even a century ago. The results have been most revealing with regard to mechanisms of vertebrate embryo development, vertebrate embryogenesis, by being able to combine these modern molecular biology techniques with some of the classical experiments that were done many, many years ago. Let's talk now about fertilization in the frog. Frogs have external fertilization, with the male fertilizing the eggs as the female lays them. The egg has polarity before fertilization. In fact, there's a dense yolk material in the vegetal pole, very little yolk in the animal pole, so it has that kind of polarity in terms of superior-inferior animal-vegetal pole. Also, there's a polarity that develops upon fertilization. At the point of sperm entry, the sperm will come in with its centrioles and start to organize microtubular structures that will set up the mitotic spindle in the area of the animal pole. Directly opposite of where the sperm enters is where really development begins, however. This is the point where the dorsal lip of the blastopore will form, where a gray crescent initially forms, and where really development of that egg begins. And we'll learn more about that in just a few minutes. But the point of sperm entry then determines the dorsal ventral polarity. The point of sperm entry becomes the ventral belly part of the embryo. That's going to be the ventral part of the embryo. And the opposite side, about 180 degrees roughly, there is a rotation that takes place which will yield the gray crescent, but approximately 180 degrees opposite that marks the dorsal or spinal portion of what the embryo is going to form. If we look at the gray crescent area, this is where the sperm has come into the animal portion of the egg, and it causes a rotation such that a gray crescent will be exposed by having some of the material underneath in the deeper cytoplasm come out into the vaginal area. There's a rotation of that animal hemisphere. This forms what is called the gray crescent, and it's basically opposite from where the sperm came in. It's 180 degrees from where the sperm came in. If we look at the structures now, here's the vaginal region of the egg, where most of the yolk is, or a large portion of the yolk. Here's the animal, the pigmented animal region of the egg, and here is the gray crescent right after fertilization. The first cleavage division after fertilization goes through that gray crescent. If it fails to go through the gray crescent, then only one side of this organism will develop, and we'll see some experiments that show that a little bit later. The other point is that if you look at the organization of microtubules in the vaginal area, about halfway through this initial phase that's leading to the first division, you see this kind of organization of the microtubules. About 70% through, however, you will start to get significantly more organization in those microtubules. At the complete cycle in the vaginal pole, the microtubules will then become aligned. The gray crescent is where gastrulation will begin, as I mentioned before, and it's where the dorsal lip of the blastopore, or the primary organizer, will form. It is where the embryo begins to develop. Also, although the xenopus does not actually show a gray crescent, it has a comparable structure because there is a rotation, and that it's the same relationship in the xenopus. This happens to be Amphibian, and you can see the gray crescent in this particular species. Let's look now at the cleavage of the xenopus egg. This is the xenopus, and it simulates where the gray crescent would be, even though you don't see it, but an area comparable to the gray crescent in this area. You can see that the first cell division is beginning in a north to south direction, and the second cleavage division does the same. The second cleavage division actually begins before the first one is finished. The third cleavage division then is equatorial. So these are meridianally placed. The third cleavage then is equatorially placed, but it's not really in the equator region. It's up in the animal pole area. It's up in the animal portion of the egg. And the reason that the division takes place up here is there's much less yolk up here to impede its division than there would be down in the vaginal region. So, as cleavage continues, you start to get more divisions taking place up in the animal hemisphere as compared to the vaginal hemisphere. And in fact, when the meridional cleavages begin and continue to go down, you get the equatorial cleavages, and you get more equatorial cleavages than you do meridional cleavages. Although the meridional cleavage will start, it will continue to go down, but before it finishes its division all the way down to the bottom, because of the dense yolk material here, cells have already been dividing up in the hemisphere. And here you can see as development progresses, there are many more cells that have formed up in the animal hemisphere as compared to the vaginal hemisphere. And so by the time you get to the Blastula stage, where a blastocele is formed, you can see that there is a significantly larger number of cells in the animal hemisphere as compared to the vaginal hemisphere. By the time you get to 16 to 64 cells up in this area, this is called the morula stage. And so it starts to look a little bit like a mulberry, and that's how it got its name from Latin. Morula means mulberry, so it's got its little bumps. Now these are
This is a diagrammatic representation, and in a minute we will see a more realistic version of what these morula stages really look like. And then it goes on to the Blastula stage, and this would be around the 128 cell stage. This is probably about the 7th division that you start to get this blastocele forming. This is a scanning electron micrograph of a frog egg during cleavage. Here's the first cleavage division, meridional cleavage going from the North Pole to the South Pole here. Right angles to that, this is the first division here. At right angles you will get a second division coming into position, and that will be at right angles to the first division, forming what's going to be the 4-cell stage. However, before it gets really to that 4-cell stage, you start to get a third division up here, and so you're actually getting more cells forming up on the top than on the bottom, as we mentioned earlier. Now, at this point, we're approaching getting to the 16-cell stage. We see now that we have 6 cells on the top. One of them hasn't quite finished dividing, so maybe we could say that's 4 and another cell in the midst of dividing, and there'll be another cell that will divide here, one of these perhaps, to really round out that 8-cell portion there, to go with the 8 cells that eventually will form here to form a 16-cell embryo. So in theory, by the 3rd cleavage stage, you should have 16 cells. Doesn't quite form that way, as you can see, because of the delay in some of the cleavages that form. However, this is really the way the design is, and it will end up then, as we said before, with many more cells forming on the top, the animal hemisphere, compared to the vegetal hemisphere. What's keeping these cells together? Well, various studies have been done immunohistochemically and biochemically, and it turns out that it's something you've already learned about, the cadherins. Various kinds of cadherins, embryonic cadherins, EB cadherins, they're called also in the frog, and that will keep these cells together as development progresses. Here we see a more advanced stage now. This is the Blastula stage. Here's the blastocele. This is a normal control embryo, and this is an embryo in which, this is Xenopus now, where the EP cadherin has been removed. The embryo where the EP cadherin has really then been, in a sense, depleted, basically the cells are starting to fall apart, and they're not forming into the appropriate structures that are required for normal development to continue. The EP cadherin's embryonic form of cadherin that's special to the Xenopus is then required for normal development to take place. Just to talk about this a little further, the mid-Blastula transition is an important point in embryonic development in amphibians, and for that matter in a lot of other of the vertebrates as well. Up to the 12th cycle or so, in other words 12th cycle of division, where you'd get about 4,032 cells theoretically, over 4,000 cells, most of development up to that point has been under the control of the RNA proteins put in the ovum by the female. The transition, however, during mid-blastula depends upon chromatin changes that begin using zygote genes. So the zygotic genes are now kicking in at about the mid-blastula transition point, and now both the male and the female genes that are in that embryo begin to function in terms of making new proteins, new enzymes, and making the materials that are going to allow development to continue. And in fact, this is the point where the cells actually become motile. They are self-motile. They're not just dividing and sort of ending up being pushed in various places through divisions and through cadherin relationships, but they also can actively move then beginning at this point. When this happens, the movements can start to form the dorsal lip of the blastopore. Here's the embryo. Here's the animal area of the embryo, the animal hemisphere. You can look at the sizes of the cells compared to the vegetal hemisphere and see that they're significantly smaller because they have divided much faster. They're what are called bottle cells down in this area that will extend from the outside to the inside of that
blastula embryo, and it starts to pull in and cause an involution of the cells. And so the cells are coming down here and they're starting to involute here through this dorsal lip of the blastopore. Here we have the animal cells, the vegetal cells, and they're moving inside. Starts at this dorsal zone. Involution will continue to move around until it's a complete circle, and the cells are coming in all around this endodermal area that will end up forming the yolk plate. Just like in sea urchins, this is forming the archenteron. It's forming like the sea urchins, except that in the case of the sea urchins, the involution was just in the endodermal area down here. However, in these amphibians, it's between the endoderm and the epidermis here, the ectoderm, and so it's really at the zone between the animal hemisphere and the vegetal hemisphere. You remember in the sea urchin, it was an involution from down here. Now, as we continue development in the amphibian, we can look at some of these cell movements diagrammatically as well as histologically with a surface type of light microscope. Here are the cell movements then during gastrulation. Here's the blastocele, so this is the Blastula stage. Early gastrulation is starting to take place here. Here's the epidermis, the animal pole, endoderm primarily down here, and here are some deep cells in here that would begin to form the bottle cells. And here are some more examples of bottle cells. I don't know if you can see them very well. If you can't hear, you look carefully in your book and you'll be able to see them, but basically they're shaped in such a way that they extend all the way from the inside to the outside of this layer and help to contract and pull these cells in. And so you start to get an involution. It's through this dorsal lip of the blastopore. And as development progresses, this archenteron gets bigger. Now this is going to form the anus. So you may recall that we are deuterostomes. So the anus forms first, the mouth forms second. The mouth is going to form over here somewhere, but it hasn't really formed yet. The hind gut is going to be formed out of this archenteron. But as involution now takes place, you're getting a layer of mesoderm formed, and some of this mesoderm will form the notochord, but it will form other components in the mesoderm as well. This will be the epidermis, and this will be the endoderm that will form certain structures that are of endodermal origin, such as the gut tube and some other endodermal structures that we'll learn about later in the course. As we get continued development, there's an anterior pole that is moving down in this direction, this anterior pole that starts up here. It's really the animal pole, but it will become the anterior pole, the anterior part of the embryo, and this will become the posterior part of the embryo. Looking histologically, these are the kinds of structures you see. The endoderm is here, epidermis is here, and you're starting to get a little bit of a blip forming where the dorsal lip of the blastopore is going to enter in and begin to form that archenteron. And as the archenteron comes in and expands, it will compress the area of the endoderm, and eventually it will get rid of this blastocele. And as we look at continuing development, the dorsal mesoderm forms here. The animal pole has formed down into this anterior region of the embryo. This will become the anterior endomesoderm, and that becomes very important because certain structures like the heart and other important organs will form out of this mesoderm. You're starting to get the yolk plug formed here. Here's the dorsal lip of the blastopore now. Here's the ventral lip of the blastopore, and there's a lateral side of the blastopore as well, and you're seeing half of it, so there would be another lateral half on this side. The embryo is continuing to form, and the yolk plug is getting smaller relative to some of the other structures. Epibole is taking place. In other words, the epidermis now is moving all the way around to surround the entire embryo. Here, it's only halfway around, but as we move down through these stages now, the ectoderm is going to form an epidermis
that will come around and enclose the entire embryo. And eventually that yolk plug will be taken into the embryo, and that too will be closed with the exception of the opening of the anus. It will be covered by this ectoderm, so the ectoderm will actually come in and line the superficial portion of the anus. This will eventually form a mouth area here. It will open up, and so we have a deuterostome, as I mentioned before, mouth formed second. And here you see it microscopically, and by this stage now, the blastocele has totally been eliminated. The mouth is starting to form in this area, and you will get neural tissues formed up here. You'll get a notochord formed up here. You'll get various kinds of linings. The endoderm will form, and endodermal structures will form down in this area right here. To look at the early movements of the Xenopus gastrulation a little more diagrammatically, this is similar to what we looked at on the last diagram, but a little more detail in terms of some of the components that are there. Here's the prospective ectoderm. Here, its involution is going to be taking place now at the blastopore lip over here. We'll get an involution taking place. There are some genes that are associated with these structures, the guscoid and the expra, being involved as factors that will cause differentiation to take place. And so now with molecular biology, we're starting to learn in these amphibians what's causing this dorsal lip of the blastopore to function. We don't certainly have all the answers, but we're beginning to, with the molecular biology, be able to apply this to the amphibians and start to really understand some of these mechanisms that for the last 100 years have not really been known, but they've been observed, and with really the idea of, gee, how does this happen? Well, we're now understanding through molecular biology exactly what some of these mechanisms are, and there's still a lot more to learn. But in any event, after the bottle cells have gone through the involuting marginal zone, called the IMZ, into contact with the blastocele wall, these IMZ cells will then involute. They'll involute into the inside of the embryo, and you start to get a second layer forming underneath that epidermis. You can see it's coming in here. Bottle cells lead the way, and it will basically cause this material that's involuting to come in and eventually form a layer just underneath the epidermis. We get a similar situation where the mouth is going to form, where there's an involution here at the end of gastrulation. There's a spreading out of the endodermal cells and an extension of some of the mesodermal cells, as well as some of the anterior endomesodermal cells. Once this happens, once involution takes place, it creates what's called epibole. And epibole is what we talked about before. It's probably better illustrated in the previous slide, where the epidermis is coming down and growing around all of the other structures. Let's look at that just for a moment, going back to the slide. So this would represent epibole, where the epidermis now has grown around all of the internal structures. And I would now like to just look at another illustration that will make this point perhaps a little bit better. Here again we're talking about epibole. Here's an early stage of gastrulation. The dorsal lip has formed, and the yolk plug is going to be here. Here's the beginning of the yolk plug. Here's the animal hemisphere area. This now is the vegetal area, but it's now becoming taken to the inner part of the embryo. The dorsal lip has expanded into lateral lips, and these lateral lips continue to have an involution taking place, followed by epibole of the epidermis going over the outside, such as we see here. This yolk plug now, at this point, has dorsal and ventral lips as well as lateral lips, and it continues this involution until it is actually totally taken inside the embryo. And along with the involution, the moving in of that endodermal material, you get an extension and a spreading of the epidermis
so that it will cover the entire outside of the embryo. At this point, you're entering into the neurula stage, where you see a neural plate forming here, and this will become the neural groove that will eventually give rise to the neural tube. Now, this is the posterior part of the embryo, so the brain is going to be up in this region. The spinal cord will be down in this region as the embryo develops. This is important, of course, in that it allows the specification of the germ cell layers. Without gastrulation, you would never get the three germ layers formed. You would not get the endoderm, the mesoderm, and the epidermis or ectoderm, so that inductive interactions could take place, and so that you would have a variety of these inductive interactions to form new types of cells, tissues, and organs that would allow a new viable individual to form.